Hi everyone. For those that don't know me, hello, my name is Rob Deaville. I manage, and it's a mouthful, the Cetacean Strandings Investigation Programme. So we're a long-term monitoring programme that investigates strandings around England and Wales, funded by Deaville, Welsh Government. Some of the team are here in the audience tonight. So we're talking about marine pollution. Let's begin, as they say, at the beginning. I want you to picture a porpoise, first of all. So this is a porpoise happily playing with little porpoise mates out in the sea. Looks nice, right? This is what should happen in the sea. But what happens when animals like cetaceans are exposed to pollution that we cause at sea? Things like chemical pollutants that end up through the food chain and affecting marine predators. Or from marine plastics that we carelessly discard and allow to end up in the marine environment. Or from the unfortunately myriad sources of man-made noise that we release at sea. So what does that mean for those marine animals in the water? And I guess, how do we learn more about these threats that impact mobile marine species? Well, stranding is one way of getting at that. That's where we come in. So strandings are, or can be, a visible manifestation of a problem out there in the big blue. And for us as stranding scientists, we can access those bodies, try and examine the post-mortem, and learn more about the threats they face in our waters. But let's be really honest, they're biased in nature. So they have to be stranded, they have to be found, they have to be reported. We have to be available and not down the pub and get to them. There are loads of biases in that data set, so really open and honest about that. But from our perspective, they're a really clear way to access information on the threats they face and give us a sense of the wider marine ecosystem. It's not perfect, but it's one of the tools in our toolbox. Over the next 80 minutes-ish, you're going to hear from 4 fantastic speakers, good friends of mine as well, about the impacts of chemical, acoustic, and physical pollution on cetaceans in our water and further afield. So, to start at the beginning again, it gives me great pleasure to introduce my good friend and colleague, Dr. Rosie Williams. So Rosie Williams has been with ZSL since 2017, having just corrected my notes, where she undertook a PhD with the Institute of Zoology and Brunel University. As a postdoctoral researcher, Rosie now works very closely within our team, CSIP, but also with the Ocean Predator Lab at the Institute of Zoology to assess the health of marine predators in relation to anthropogenic stressors such as noise and chemical pollution, which we're talking about today. Rosie's current interests lie in developing new methods to understand the impacts of chemical pollution on wildlife health and exploring ways to better characterize and understand the cumulative impacts of anthropogenic stressors, if I can say it properly. So, please welcome Rosie.
Thanks very much, Rob. So today, what I'm going to do is give you a brief introduction to marine chemical pollution, and then I'm going to talk about why marine mammals are an important group to study chemical pollution. I'm then going to talk a bit about the impacts of chemical pollution on marine mammals and some examples of that using the Strandings data in the UK. And then I'm going to finish by talking about what we can do to try and tackle this really important issue. So as I'm sure you're all aware, the chemical pollution crisis severely threatens human and environmental health. And there are lots of different types of ocean pollution, so ranging from things like plastic waste and the chemicals that are associated with that, things like manufactured chemicals and flame retardants, and then things like mercury that derive from combustion and metal mining. But it's important to say that alongside there being a real breadth of different sources of chemical pollutants, more than 80% of the pollution, chemical pollution that's in our ocean is derived from land-based sources. So this is really an issue that we need to tackle by better regulation on how we're managing chemicals on land. And so marine mammals are some of the most polluted species in the world, which makes them really important to study to understand the impacts of chemical pollution. So they're particularly vulnerable to accumulating high levels of chemical pollutants for a few reasons. So they're long-lived, which means these chemicals that bioaccumulate in them over their lifetime can accumulate to high levels. They also feed at high trophic levels, so at the top of the food chain often. And so these chemicals biomagnify and concentrate through the food chain, meaning that some of these types of pollutants accumulate in high levels at the top of the food chain. They also have a thick layer of blubber or fat, whereby some of these chemicals, which are lipophilic, so they bind to fat, can accumulate to high levels. So in terms of wildlife, these are some of the most highly exposed species in the world. And then the second reason why they're really important to study is that they're often considered to be really important indicators of marine ecosystem health. So if we can understand what's going on in these animals at the top of the food chain that are long-lived, then we can say a lot about the health of our oceans generally by studying this group. So the first thing I wanted to do was just have a shout out to how brilliant our UK data is in terms of being able to understand chemical pollution in this group. So to the best of our knowledge, we've got the world's largest cetacean toxicological database. So we've got toxicological data on over 1,300 individuals, which is a really fantastic data set. And that's because the program's been running for such a long time, and also it's something that's really prioritized within the program. So this figure was from a paper that we published in 2023 that looked at 6 different chemical pollutants across 11 species with varying sample sizes. And I wanted just to point out a figure on the left. Each of these dots is colored by the species and then sized by the pollutant burden. And the thing I want to pull out are the sort of big green blobs and the big orange blobs, which are killer whales and our bottlenose dolphins. And that's just really reiterating the fact that the species that are at the top of the food chain and the longest lived are the most susceptible to being exposed to high levels. And then the figure on the right are the harbour porpoises that Rob talked about at the start. And they're really our sort of flagship species for being able to do powerful statistical analysis to ask important questions about chemical pollution in the UK. So they're the UK's most abundant marine mammal species, cetacean species, and they're really widely distributed around the whole of the UK, which allows us to look at spatial trends of contaminants and also look at temporal trends, but also do this powerful statistical analysis where we can try and relate their exposures to markers of health. So they're really important species for us to be able to understand what's going on and then extrapolate to potentially more vulnerable species for which we have lower sample sizes. The other thing I wanted to mention is how we decide which pollutants to study. So at the last inventory, I think there are around 350,000 different chemicals and mixtures of chemicals produced by humans. So it's really important, obviously, to target the right chemicals to test within our marine mammals. And in the early days of the project, there were a few pollutant groups that were really prioritized and thought to cause the biggest issues that are highlighted on this slide. And we saw really nice declines in the majority of these pollutants that corresponded with the correct regulation coming into place that could then lower emissions, and then we'd see really nice declines in wildlife too. The exception to that were the PCBs, which are the purple in these figures here. And unfortunately, we just haven't seen the declines that we would have that are similar to the other pollutants. So they seem to be declining at much lower rates. And so we've really prioritized this chemical group within the analysis that we do. And this is some work where we just kind of wanted to reevaluate that, and we looked at a thing called critical risk quotient. Essentially, we just wanted to see how much of the risk from each of these pollutant groups was attributable to each pollutant, and we looked at 2 different endpoints. So we looked at impacts on the hormonal system, so disruption to that, and then we looked at impacts on the immune system. And the takeaway here is, across all the species that we looked at, the purple is dominating both of these sort of risks that we looked at, and that's the PCBs. So we've continued to prioritize those in the UK, but we do also look at things like heavy metals and other emerging contaminants that you might have heard of, like PFAS, just to sort of keep an eye on what's going on. So first thing I'm going to talk about using our UK data is looking at trends over time and space, and talk about a couple of examples. So I've mentioned these pollutants known as PCBs, and what we found in harbour porpoise is that they are decreasing, which is good news for sure. But unfortunately, these rates of declines that we're seeing are not what we'd expect or hope for, and are much slower than other pollutants that we've seen. But they have, in the most recent year that we looked at, fallen below levels that are deemed a toxicological threat. So that's the green lines and the red lines on this figure here. And that's where we think that these pollutants are likely to have physiological impacts. So that's good news in that we're seeing that they've fallen below that level, but they're not falling at the rate we would have hoped for. And this is potentially evidence of long-term preservation within the population and potentially continued environmental contamination from things like diffuse sources. The other thing that I wanted to call out on this figure here that you might have noticed is we see different rates of decline in different regions of the UK. So you can see that in Scotland, we had a bit of an increase around the year 2005, corresponding with decreases in other areas. And we think this is probably evidence of short-range atmospheric transport, whereby these types of pollutants that are persistent and can undergo long-range long-range transport move from areas of high concentrations to low concentrations, and also from lower latitudes to higher latitudes. So we're seeing evidence of this in the UK, but it's also important to note that this is happening on a global scale. So we're seeing these types of pollutants in pristine ecosystems like the Arctic and Antarctic, sort of hundreds of miles away from where these chemicals are produced and leached into the environment, but they undergo this transport via the atmosphere and the oceans and can pollute these really pristine ecosystems. So I think that just highlights that we really need international cooperation to try and tackle these issues of chemical pollutions, because they ignore borders and are really persistent, and so can cause problems for a long, long time if they're not regulated for properly when we discover that they're an issue. The other thing that I wanted to talk about is some more recent work we've done looking at heavy metals in harbour porpoises. And we looked specifically at mercury because that's known to be the most toxic heavy metal. And we found, unfortunately, that mercury concentrations appear to be increasing in our UK harbour porpoises. And it's quite an interesting finding because there's also evidence of this happening in other species and locations, despite the implementation of the Minamata Convention, which aimed to reduce mercury pollution. So we aren't necessarily seeing the declines that we'd hoped for, even though there might have been a reduction in emissions. And there are a few explanations that are out there at the moment, and people aren't necessarily sure why this is happening and why we're seeing this, but some of the sort of hypotheses have involved climate change, altering the rates of deposition and bioaccumulation. Also things like changes in ocean currents. So mercury, for example, will quite often be locked up in the deep ocean. And if we have changes in ocean currents associated with climate change, that can then increase availability and mean that it's available again in surface ocean food webs. So this is something that we're not just seeing in the UK, but it's unfortunate that we find this because our previous analysis around a decade ago had shown that mercury levels were decreasing. So now that we've talked about trends in the UK, I think it's really important, as well as looking at spatiotemporal trends, to understand if there are any links with markers of health that we can then determine from our Strandings data to understand if there are potential impacts in the population. This is really difficult to achieve because obviously the animals are already dead. So a lot of the biomarkers that we might use to understand about the health of these animals aren't viable. The other issue that we have is that these pollutants tend to cause quite chronic effects. So there's not really ever a smoking gun when we carry out a post-mortem whereby we can say with certainty that these animals have died from chemical pollution. But there are some techniques that we can use, and because of that harbour porpoise data set that I mentioned, we can do some statistical analysis to try and understand if there's an impact. So when we talk about PCBs, there are real two modes of toxicity that we talk about with these pollutants. So they can have impacts on the reproductive system, and they can have impacts on the immune system. And if you think at the population level, that can lead to reduced births and an increased number of deaths, and that can be a real double whammy on the population in terms of impacts. So the figure on the left is some work whereby they've modelled calf survival in the first 6 months and related that to the maternal concentrations of PCBs in the blubber, and you can see that there's a negative relationship there. Within our own UK database, we've also found that our animals have reduced pregnancy rates in comparison to less polluted populations. The other thing that we've looked at is lactational transfer of these pollutants. So unfortunately, the reason why you see this relationship is because these pollutants that I mentioned are lipophilic, so fat binding, which means that they also bind to the milk. And so mothers can transfer and offload their pollutant burdens to their calves. They receive a large dose of these pollutants early on in their life, and that can reduce their survivability. The other thing that I wanted to mention is these immune system impacts. So this is some work in vitro, so in a laboratory, looking at immune system response and how that relates to PCB concentration. And you can see there's a negative relationship there across several marine mammal groups. So this has been proven in the lab, but we also want to see what's happening in the real world environment in these animals. And that's where the Strandings data is really helpful to be able to do that. So when we want to investigate things like immune system suppression, what we do is we look at infectious disease as an endpoint for that. So what we do is we do a case-controlled approach whereby animals that have died from infectious disease that have stranded, we deem as cases, and animals that have died from something like trauma, so are caught in fishing nets or bycaught, we take as controls. And then we can do some statistical modeling to try and infer associations between the pollution and how they died and whether they're suffering an immune system impact. And this is a really similar approach that's used in a lot of human health studies whereby you can't do laboratory testing. So this is the kind of thing that would be used to find associations between lung cancer and smoking. And so when we did this using our UK Strandings data, we found that there was a significant association between animals that had died from infectious disease having higher concentrations of pollutants in their blubber. And the figure on the right here is just showing you that relationship between an increased risk of death from infectious disease and the PCB blubber concentrations. And so we found that in the most recent year that we looked at, which was 2017, we found that the mean concentration of PCBs in our UK harbour porpoises led to an increased risk of infectious disease of 41%. So it is likely that the level of pollution they're exposed to is causing an increased number of deaths within the population. So I think it's also really important to just talk about specific animals as well to try and understand and relate a little bit more about what this looks like in the real world. So I just wanted to talk briefly about a case study. This is a killer whale that stranded in 2001. It stranded on a sandbank in the middle of the Mersey estuary, and Rob tells me it was a very glamorous trip when they chartered a helicopter. I don't think it was glamorous because they were in the middle of a sandbank and it was really hard to access, but they did have to charter a helicopter because it's really important that we get samples from species like killer whales. They're few and far between, but we know that they're exposed to some of the highest levels of PCBs and other pollutants. So we really prioritized going and getting samples for this animal. It was an adult male around 6 meters long, and it was quite debilitated. So it had worn teeth, one with an abscess. It had parasitism in multiple stomach chambers. And although, like I said, we can't necessarily say there's a smoking gun, the levels of PCBs within this animal were 190 milligrams per kilogram. The thresholds that I was talking about before, the lowest proposed threshold for toxic effects is 9 milligrams per kilogram. So really significant burden of PCBs in this animal. And just to highlight here that the bar on the far right is the concentration of PCBs in this animal, and you can see that that's higher for the mean average of killer whales in Europe, bottlenose dolphins, and then harbour porpoises. And this is some work done by Paul Jepson in 2016 looking at concentrations in various marine mammals. So really high levels in this animal. If you want more information on this specific case study, but also other case studies, then Rob's pulled together a really nice Padlet. I think there's a link somewhere we'll talk about later, but you can read a lot more on there. But I just wanted to call out that this is obviously not a nice way for these animals to go. They're often really debilitated. The sort of immune suppression aspect of these pollution means that they really do suffer in the environment. So I think it's important just to highlight that. I wanted to finish on a sort of more positive note. So this is a very cute grey seal pup. And I wanted to use this to talk about something called the Baltic seal disease complex. And this kind of was first talked about in the mid-1970s, whereby these animals had significant uterine obstructions, so obstructions in their uterus, things like occlusions and tumours that were impairing reproduction. And you can see around the mid-1970s in the figure on the left that they have really high percentages of these tumours and occlusions. And that corresponds with really low pregnancy rates, so they were really impairing reproduction in this population. That figure on the left, the decline in uterine obstructions, corresponds so nicely with this decline in PCB concentrations, and it was hypothesized that the levels of PCBs they were exposed to were causing these tumors and reduced pregnancy rates. And you can see that as the concentrations of PCBs declined, you had this lovely increase in the pregnancy rates that really nicely corresponds and shows that these animals, if the right mitigation is in place and the right regulation is in place, they recover really quickly, and there is hopefully a positive message in that if we take the right action, then these animals will recover and we see reproduction and they manage to reproduce and the population can recover relatively quickly. So the final thing I wanted to mention was what can you do about this issue? It's really tricky, and I think a lot of it lies at the government and international level, like I've said, because of these pollutants and their crossing boundaries and us needing international cooperation to properly regulate. But on an individual level, there are things you can do. So things like choosing safer products, things that aren't sort of harsh, like cleaning products and pesticides, cutting your plastic waste. So there's a lot of chemicals associated with plastic production and plastic itself. Disposing responsibly of chemicals, so things like your medication and paint and chemicals not down the drain, and then supporting policies, joining citizen science and raising awareness. And then at the system level, I think it's really important that we regulate these chemicals more proactively. So often it's quite reactive once we've proven they're harmful and persistent in the environment. I think we need better regulation that's proactive to sort of stop us having to clean up the mess several decades later. Also, things like better waste management are really important so that we don't have emerging chemicals entering into the environment. And then things like global monitoring, also really important. So projects like CSIP, the Cetacean Strandings Investigation Programme, really important to monitor impacts from chemical pollution, but also on a global level, having things like human health monitoring and environmental monitoring are really important to know where we're at in terms of what pollutants are causing issues in the environment. I'd just like to finish by thanking Paul, who was a supervisor in a lot of my PhD work and inspired most of, or all of the work that I've done today. He was a huge pioneer in this space and within CSIP, so thanks to Paul for all his support in my career. And then I'd just like to acknowledge the huge amount of people that are involved in collecting this data. I'm sure you can imagine it's a mammoth effort over almost 35 years at this point, and I'm hugely indebted to them all for allowing me to carry out this research. And thank you all for listening and coming along today.
Thanks, Rosie. Brilliant but depressing thought. We've run out of time for questions, but before you go, I'd say thank you very much. Fine bottle of my favourite Pinot Noir.
Oh,
so
lovely.
thanks, Rosie. Stay for that, right?
Yeah.
Thank you. Thanks, Rosie. Okay. So perhaps any questions for Rosie? We can save those for the end when we have the Q&A. I'm going to introduce the next speaker. It gives me great pleasure again. Lots of pleasure tonight. Introduce my good friend and colleague, Dr. Sarah Nelms. So Sarah is a senior lecturer in marine vertebrate ecology and conservation at the University of Exeter Centre for Ecology and Conservation, based at the Penryn campus in Cornwall. Woo! Where was I? Okay, for the woo. Sarah takes a multidisciplinary approach to understand the issue of plastic pollution, for example, by using citizen science data to map the abundance and distribution of litter in marine environments and sifting through marine mammal guts, yes, I can testify, definitely guts, to look for microplastics. She's lead supervisor to 2 PhD students whose projects are possible due to collaboration with CSIP, with me apparently being involved as well. Yes, that is also true. The projects use Strandings data and samples to understand marine mammal bycatch in southwest England and plastic ingestion by marine megafauna, and they're both fantastic students as well. So Sarah, thank you very much. Thanks, Sarah.
Thanks, Rob. So I actually just want to say a huge thank you to the ZSL team for inviting me to be here and speak at this great event. It's a real honour to be here. So today I'm going to be talking about a physical pollutant in the form of plastic pollution and the kind of impacts it can have on marine mammals. So over the last 2 decades, the number of marine species impacted by anthropogenic debris has more than trebled. Plastic pollution is persistent, ubiquitous, and it's increasing. And for marine mammals, plastic pollution presents 2 main issues through entanglement and ingestion. Marine mammals are arguably one of the most studied and perhaps the worst impacted marine species groups for entanglement. It seems that the majority of entanglements is caused by so-called ghost gear, which is abandoned, lost, or otherwise discarded fishing gear, that continues to drift in the ocean and catch animals indiscriminately. This rather sad image at the top there shows a pregnant minke whale that stranded in Orkney in 2019, and it was attended to by the Scottish Marine Animal Stranding Scheme. And as you can see, it's got quite a lot of fishing net trapped in its mouth, and actually it was completely jammed within the baleen and was preventing this whale from eating and moving properly. But the actual rates of entanglement are really difficult to assess. The signs of entanglement in plastic pollution can be very similar to those of bycatch, which is when an animal becomes entangled or trapped in active or operational fishing gear, as opposed to ghost gear. But land-based sources of plastic pollution can also cause entanglement, and things like frisbees, as they're known, or flying rings, seem to be a really big problem for seals because they poke their heads through the hole and it gets stuck on their necks. So the impacts of entanglement are a huge welfare concern. Entanglements can cause increased drag and reduced swimming ability, which in turn affects the animal's ability to feed, escape predators and other threats such as vessels. And the site of entanglement can become severely injured and often results in loss of limbs or increased risk of infection. This is another female minke whale that stranded on the coast of Yorkshire in 2020 and was attended by Rob and the CSIP team. And as you can see from the picture, she was severely emaciated, and it was likely due to long-term chronic entanglement from around the tail, and also the tail fluke had become partially severed as well. And obviously this probably took quite a long time and resulted in a very painful and slow death of the animal. Aside from the individual level of effects of entanglement in plastic pollution, the population level effects are really difficult to assess, likely because they're just really hard to identify, but they also work in synergy with other stressors. We can expect, however, that the likelihood of population level effects increases when you've got species that are already of conservation concern overlapping with areas that have high concentrations of marine debris. And a prime example of this is the Hawaiian monk seal, which Francis will be talking a bit more about later. So obviously, strandings can tell us a lot about the impacts of entanglement, but I just wanted to highlight a case that kind of demonstrates the obvious and not so obvious injuries it can cause. This is a female grey seal that was first encountered in Cornwall in September 2018 with a large encircling neck wound consistent with monofilament net entanglement. She was then re-sighted about 1.5 years later because she became stuck in some rocks and had to be rescued by the team at British Divers Marine Life Rescue as the tide was coming in. So they rescued her and attempted to rehabilitate her, treating her neck wound, but unfortunately she deteriorated and was euthanized 9 days later. Following this, a post-mortem examination was carried out by James Barnett at the Cornwall Marine Pathology team, which revealed that she obviously had a severe neck wound, but she was also in poor nutritional state and was very small for her age. And interestingly, her liver was really large and pale and had signs of past and current hemorrhaging. And it was thought that this chronic inflammation caused by the long-term entanglement had led to this liver damage and subsequent poor growth rate and poor nutritional state. So moving on to plastic ingestion. And I just want to start here by talking about macroplastics. And these are the large pieces of plastic, generally larger than 5 millimeters in size, although usually bigger. So these are the bottles, bags, ropes, etc. And there's a variety of reasons why marine vertebrates might consume these items, including active selection whereby animals mistake plastic for food. This is likely driven by sensory cues such as sight and smell, but for the deep diving odontocetes or the beaked whales, the toothed whales that use echolocation to forage, misperception of acoustic signals could be a key driver in plastic ingestion. So a recent study found that some marine debris items have really similar acoustic signals to those of whale food items, and this could explain why species like the sperm whales and the beaked whales seem to be more susceptible to plastic ingestion and becoming compromised by it. So the impacts of ingesting large pieces of plastic are usually quite obvious. They include injuries and blockages to the digestive tract. This can lead to dietary dilution and malnutrition, and it can also mean that animals are less resistant to stressors like disease. And as Rosie mentioned as well, plastic contains chemicals on it and within it that are added during production or adhere to the surface from the environment. And ingestion of plastic can mean that animals are exposed to these chemicals, and as we've heard, they can have impacts on reproduction and immune system function. So this is an interesting sort of case study that, again, Rob from the CSIP team attended. It's interesting for a variety of reasons. So firstly, it's the first ever sperm whale stranding in Wales. And it was also a very young animal, so it was out of habitat. An animal at this age should be much further south in warmer waters. Interestingly as well, it had lots of marine debris within its digestive tract. So there was rope and other plastic debris in the cardiac stomach, which is the first chamber within the system. And there was also this blue sheeting that ran from the cardiac to the fundic stomach, which is the next chamber along. It was nutritionally compromised, likely as a result of all this debris in its digestive tract, which likely also led to its death. So moving on to microplastics, which are classified as being less than 5 millimeters in size, but they're usually much smaller than this in the samples that I kind of work on. They can be purpose-made for things like cosmetics and industrial abrasives, or they can be formed from macroplastics that have broken down through UV from the sun and wave action while they're in the environment. And marine mammals can ingest them from two main pathways. Firstly, direct ingestion, which is when microplastics in the seawater are directly consumed, and this is usually the case for the filter feeders. And then we've got indirect ingestion or trophic transfer. And this is when microplastics are present in animals at the base of the food web, so things like zooplankton and small fish, and these are in turn consumed by marine mammal predators, and the microplastics move up the trophic levels within the food web. Of course, some animals are also likely exposed to both direct and indirect ingestion where the microplastics in the prey and in the seawater. As I said, filter feeders seem to have a higher propensity to ingest microplastics, and we estimated that some baleen whale populations are exposed to around 3 million microplastics per day because of this kind of double whammy. So to explore the extent of microplastic ingestion in UK waters, I worked with the Scottish Marine Animal Stranding Scheme and CSIP to analyse the digestive tracts of 50 different individuals from 10 species of small odontocetes, mainly dolphins and porpoises, as well as some seals. The coloured dots on the map just correspond to the species and where they stranded. And I found nearly 300 microplastics across the 50 animals that I examined, and every single animal had at least one microplastic in their digestive tract. And these are just some examples of the kind of microplastics I found. I also found that animals that died of infectious diseases had a slightly higher number of microplastics than those that died of trauma or other causes of death. We don't know what the biological significance of this is, or even if there is one, and we're trying to obtain funding to find out whether there's actually any kind of health impacts from microplastic ingestion for these marine mammals. So nylon was the most common polymer type that I found within these animals at 61%, followed by PET and polyester at 10%. And these are found in everyday items like ropes, clothing and textiles, and food and drink packaging. So in terms of understanding the implications of plastic pollution for marine mammals, there's a whole array of different questions that we could be asking, but I'm just going to prioritize a few, I'm just going to highlight a few priorities in terms of research and next steps. So the first is identifying which species or populations are most at risk from interacting with plastic pollution. We've conducted a spatial risk assessment for land-derived litter in the North Atlantic for cetaceans, and it combined metrics for hazard, exposure and vulnerability to provide a score of 0 to 5, with 5 being the highest risk areas. And although it's quite a crude sort of calculation based on limited data, we were able to highlight 5 high-risk zones within the North Atlantic, and these were the eastern coast, the Atlantic coast of the US, the Gulf of Mexico, the Azores archipelago, the French Atlantic coast, and the waters around the UK. So moving forward, this could be replicated on a species level at a global scale and for all types of plastic. And one of the biggest questions relating to plastic pollution is, what are the health impacts? Obviously, we know that ingesting or becoming entangled in macroplastics can cause injuries and mortality. But what about the smaller microplastics and even smaller nanoplastics, which are less than 1 micron in size? And the key concern with these is that they're so small that they can translocate across the gut wall into the bloodstream and make their way to organs where they could cause disruption. And a recent study by Greg Merrill at Duke Marine Lab found microplastics in the blubber, melon, acoustic fat pad, and lungs of marine mammals, so it definitely requires further research. And it could also mean that inhalation is an exposure pathway that we haven't really considered that much, but could be key to how these microplastics enter the bodies of marine mammals. As I mentioned earlier, chemicals that are present on and within plastic can be released when they're ingested, but we don't really know how plastic exposes these animals to those contaminants, as aside from their normal kind of environmental and dietary input. And plastic in the environment can be colonized by a whole range of bacteria, and ingestion of plastic could expose animals to increased levels of pathogens. And again, we don't know how this can affect animal health yet. My last point is more of a practical one, but it's really important if we're to kind of answer some of the questions that I've just raised. And this is, how do we go about sampling the larger animals in a reproducible and representative way, especially for microplastics? Currently, only about 8% of studies have attempted to assess the microplastic ingestion in large whales. And there is a good reason for that. It's like looking for a needle in a haystack. Just for to demonstrate the point, this is me in the lab with a harbour porpoise intestine, which is the UK's smallest cetacean, versus Rob and his team providing scale for the sperm whale intestines, which could be hundreds of metres long. So one way is to subsample. This is myself and a student sectioning off the intestines of a juvenile minke whale into 20 centimetre sections, and we were sampling every other section. In this case, we could lay out the entire length of the intestine, measure it, and work out the proportion of the animal that we were actually sampling. We could also control and monitor to some extent the sources of external plastic that might contaminate our samples. But this was actually quite a small animal. What happens when an animal is so big that it has to be sampled in situ, such as this fin whale, 16-metre fin whale that stranded on the coast of Cornwall a few years ago? And this is my PhD student, Emma Hunter, and Rob's PhD student, who is currently working on trialling various real-life and theoretical ways of sampling these ocean giants. So watch this space. So I just want to end by acknowledging all of the people that have worked on this research. It obviously wouldn't have been possible without them. And just to highlight the fact that none of this would have been possible without the input of the stranding schemes. Thank you for listening.
Thanks very much, Sarah. We have got time for one very quick question. If anyone's got a burning question to ask Sarah, please put your hand up, or maybe online. And if not, cogitate. Oh, there's a question here from Ethan.
Hi, Sarah. Thank you for that talk. I was just wondering, if you did get the funding, how would you investigate the relationship between plastics and infectious diseases?
Oh, that is a tricky one. Yeah, so we've talked about before potentially kind of looking at the viruses and things that are on the plastics that we extract from the stranded animals. I'm not sure if it's possible, but I guess that would help us to understand what's there, but we could also look at the extent of infections within the animals and the extent of microplastics present within their digestive tracts as well.
Okay. Before you go. Thank you very much.
Thank you. Thank you.
So we're going to go home tonight. Thanks. Thanks, Sarah. Okay, moving on. So quite keen to keep time for questions at Q&A at the end. So I'd like to introduce our next speaker, again, a good friend, Nikki Taylor. Nikki is a marine conservation specialist focused on protecting vulnerable marine species through science-driven policy and strategic collaboration. With over a decade of experience, she's led national and international assessments on marine mammal population status, represents JNCC and the UK international forums, and has advised on the implementation of marine protected areas. She works in collaboration with cross-sector partners to develop mitigation strategies. Why is it doing that? Not me. For impacts such as bycatch and climate change. Nikki is committed to bridging science and policy to ensure resilient marine ecosystems and lasting conservation impact, and she's a good egg. Thank you, Nikki.
Thanks for that, Rob. Who doesn't like to be a good egg? Hello, everyone. I am Nikki Taylor, Senior Marine Species Advisor at JNCC, and I'm going to talk about marine mammals and noise. So one of the first questions I normally get when I say I'm from JNCC is, who are JNCC? So I'm going to very briefly say who JNCC is. We're the statutory nature advisor to the UK and devolved governments. We work as a coordinating body. We work very closely with the country nature conservation bodies, and we work with scientific evidence and advice to support nature conservation. So, I just wanted to quickly introduce how marine mammals rely on noise. So it's vital for most aspects of their behaviours. They rely on it for communication, for navigation, and for foraging. So in the image here, most people are normally quite familiar with how bats function with echolocation. Marine mammals have a very similar way of doing things, producing sound, which gives them a signal for what's in front of them, and then that's received back, and they can interpret what's there so they can find their prey, avoid obstacles, and that kind of thing. And then the bottom image there is how we've interpreted that as well for our own needs. And yeah, for many of them, sound is both their eyes and their ears, so it's a super important part of being able to function. So the underwater soundscape is quite complex. There's both natural and anthropogenic noises. So some of the natural sounds, if any of you are divers, you might have experienced some of these when you've been in the water. So there's things like the weather changes, so heavy rain, thunderstorms, underwater earthquakes, and then general water movement. And then there's also the noises that the animals are making, so the vocalizations of the whales, noises from snapping shrimp, which are surprisingly noisy for something so small. And then we have the human-made noises, such as that one, which I didn't actually ask to play. There we go. That's the noise from a ship's engine. And we have pile driving, so when they're installing offshore wind, that's the sort of rhythmic hammering in of the piles into the seabed. And then the last one here is underwater explosions. So after we've been dropping all the lovely bombs, a lot of them end up on the seabed unexploded, and there's a process of trying to deal with that to make navigation and operations in the water safe. So there's a process for exploding those. And then within this, there's the two variables of frequency and pitch. Yeah, so the frequencies and the decibels are the two aspects that really impact how much that's influencing whether animals can communicate underwater. Okay, so what are the impacts of all this underwater noise? So we have some short-term effects, things like startle responses, which is essentially a stress impact. Then there's decompression sickness, or something that mimics what you see in humans as decompression sickness, which I'll talk about a little bit more in one of the case studies. Then there's altered diving patterns. Sometimes there's temporary hearing loss or impairment from impulsive sounds or prolonged exposure to sounds, and behavior disruptions as well. So then we also have the longer term consequences. So permanent hearing loss, if you have strong exposure to an impulsive noise, that can cause permanent hearing loss. And then that, of course, has knock-on effects on their ability to forage, feed. And then things like habitat abandonment, impacts on reproductive rates, and eventuality could be mortality as well, just from having some of these impulsive sounds in the water columns. So what can Strandings tell us about noise? It reveals some hidden stories that we can't really see through many other sampling methods and ways. It gives us access to unique information, and it's a critical part of the broader puzzle on how we can understand the impacts that noise is having on our marine mammals. So post-mortem analysis allow us to investigate actual causes of death, but also other influencing factors, and impacts of pressures more generally that we might not be seeing through other monitoring methods. The impacts of noise can manifest in multiple ways. So some of these we can only see by actually going inside an animal and exploring what's happening to internal tissues and organs. And this includes things like establishing decompression sickness, or the similar aspects that are seen in some of the marine mammals, and also ear damage. And that image there is part of an ear bone, I believe, from a minke whale that's stranded in the UK. So this is one of the case studies that I wanted to go through. So this is from a Risso's dolphin, an adult male. Risso's dolphins are really deep diving species. They can go down to great depths, and they feed mainly on cephalopods, so things like squid. This field necropsy was completed by Dr. Paul Jepson, who's already been introduced, and also Rod Penrose, both of whom are Strandings legends. And this post-mortem confirmed chronic gas embolism. and that was in multiple organs. So this is quite an unusual find because the understanding is that marine mammals are well adapted to working in this environment and aren't subject to the same issues that we see in humans. But this is now obviously being seen through this case in particular, but also several other cases that are being explored. So the cause of this is unclear. It could be natural or human, but the fact that we have this understanding that this is indeed happening in marine mammals gives us that little bit of the puzzle that helps us to think a little bit more about how we might need to mitigate our own behaviours to try and make sure that we're not the cause of these types of eventualities. So some of the similar cases in Canary Islands, for example, highlighted possible links with impulsive noise, but also potentially lethal interactions with prey. So just have an image of Rizzo's dolphins and squid having battles in the depths of the ocean, but it just kind of gets you thinking, and it shifts the way that we view cetaceans and how we think about pressures and pressures mitigation. So the next case study is common dolphins. So this was a case where there were several reports coming in of a group of animals that were quite close into shore, and they were displaying unusual behaviour. So they were milling around, looking a little bit agitated, a little bit confused. There was a large-scale rescue to try and drive these animals back out so that they weren't stranding. But inevitably, we had 26 that were recorded dead from that event. So there was some exploration conducted as part of this to try and establish whether there were any sources of noise that might have been occurring that could have been happening at the same time and in the same area that could have had links. And because of the spatial overlap and at the time that they happened, we also had evidence of historical connections with noise leading to similar eventualities elsewhere. And of course, absence of anything else that there was any evidence. The probable cause was some form of impulsive noise, probably through military activity. Then the final one is this unusual mortality event for beaked whales. So there were 107 whales recorded dead stranded across the UK and the Irish Atlantic coast, and you can see in that graphic where the spread of the strandings happened. So these were predominantly Cuvier's beaked whales, so another really deep diving species, spends a lot of time underwater, and then also a small cluster of northern bottlenose and a few other species as well. This was also reported in Iceland and Faroes, so there's potential that this was much broader than initially thought. But there was limited potential for necropsy because of the condition of the animals. But there's still opportunity for exploration, given that we had this record of where all these animals were stranding and when. So this then leads to another form of use of the strandings data, so drift modelling. So you can see in the graphic there that there's quite a few things that impact how a carcass behaves in the water. So they can either be on the surface, sometimes they're below the surface, sometimes they sink down to the bottom. But if they are on the surface, then there's things like wind, waves, predation, tides, currents, all these kind of things that impact where these animals end up. So by using this information, you can start to think about potential sources where you might have had the beginnings of this mass stranding event. So the Scottish Strandings team teamed up with some researchers at SAMS to try and fit some models to see if they could come up with anything that worked that might be able to give an indication of where the point of source was. And this ended up being a relatively good fit model, which indicates that actually there was quite likely a point of origin for that strandings event, which then, I mean, we don't know what the actual cause was, but it feeds into discussions on mitigation, because if it did happen to be a human-derived sound, then that's something that we can work with when we're thinking about how to change behaviours and limit our impacts. So all of this, the long-term monitoring leads us on to be able to think about how we can reduce that impact. So we work a lot on the monitoring and mitigation side of things, and there's different aspects that we can do to try and reduce the impact of noise on our marine mammals. So you can adapt some of the designs of our technology, so things like electronic ships, for example, to reduce engine noise. We can deploy noise dampening systems when we are making noise. So that image there on the side is of a bubble curtain. And that sort of contains the sound, or at least dampens down any sound moving outside of that area, so that you're reducing that impulsive noise impact. And then there's operational measures, things like speed restrictions to reduce engine noise. You can do operational measures such as having observers on board, so that you can actually see real time where the animals are, and if you're going to be doing something that could impact, when you have to stop or start, depending on what's in the area. And then monitoring collating sources of noise. So the JNCC holds the Marine Noise Registry, which that hosts all the sources of impulsive noise. So having all this information enables you to build a picture of what's happening, and then that leads you to be able to work out how best to go about mitigating the impacts. Okay, so just to close, so we know sound is critical to marine mammal survival. We know that the marine soundscape is changing. It's generally increasing year on year. Things like cruise ships are becoming more and more prevalent. We have targets to meet in terms of putting renewable energy in place, so that's increasing the amount of pile driving and activity at sea. And these long-term monitoring aspects, such as the Strandings programs, are providing really critical information to help understand what the impact is to drive these mitigation methods and solutions. So yeah, we know that Strandings have capacity to contribute insights, and they have done on many occasions. So I just wanted to mention as well, this website DOSIT, it's the Discovery of Sound in the Sea, and they've got some really great sound bites and things that you can go on there and listen to if you're interested in hearing humpback whales singing or pile driving or whatever you fancy. There's loads of really great sound bites on there that you can get a real idea of what is going on in the ocean and what these animals are experiencing. And there's a huge amount of information as well, if you're interested. And then I just wanted to thank the Strandings teams in particular for helping me pull together the most appropriate case studies. But yeah, that's me. Thank you.
Thanks very much, Nikki. We also have time for one, really quick question, if that's okay. Is anyone in the audience, lady at the back there, and you hold that question for next time? Sorry, just the lady at the back there. Thank you, sorry.
I don't know if this is a very silly question, but seeing as the whales and the dolphins go swimming around the world and stuff, are they affected by stuff that happens on land, like, the bombs in Gaza, like the 10 Hiroshima type of bombs? Would they have been affected by that sound? And also, when it rains and the stuff goes into the sea from the land, is that going to have some long-term effect?
So in terms of the sound that's been created on land, there's limited interplay between the animals in the ocean. But there will be fallout from those types of eventualities. So there'll be potentially more ship activity. So the sonar sounds, engine noise, potentially some of the louder noises might be carried out in the kind of coastal areas. So there is potentially overlap, but everything that's happening on land is going to have limited impact compared to what's happening in the ocean. And I think for the second part of your question, were you meaning about the sort of pollution element of things flowing in? Yeah, I mean, I'm probably not the best out of this group of people to answer that one, but yeah, I think pollution that's coming from land is one of the key sources of what's happening in the ocean. So those coastal species that are spending more time around coastal areas where you're getting that runoff of contaminants are going to be the most affected.
We'll come back to your question at the end, if that's okay. Thank you. Thank you very much. Oh, hey, sorry. More wine. More wine.
Never have too much wine.
Exactly. Thanks, Nikki. Thanks again. So, final speaker for the evening, and it gives me huge pleasure to introduce one of my personal heroes, Dr. Francis. Yeah, sorry. Dr. Frances Gulland is a veterinarian specialising in marine mammal medicine and pretty much everything else. I found out tonight she speaks Italian as well, just as an aside. She chairs the US Marine Mammal Commission, is a research associate at the University of California, Davis, and worked for 25 years at the Marine Mammal Center in Sausalito, California, where her focus is on treatment of stranded marine mammals and research into the causes of diseases in these animals. She also started her work here as a wildlife veterinarian at the ZSL in 1985. Yeah, okay, so some time ago, and we were talking about the state of the chairs here, whether they were here.
Chairs are the same.
Same chairs, same. Thanks very much, Frances.
Thank you, Rob, and thank you so much for inviting me back, and the chairs are the same. So we've heard some really fantastic talks about really detailed studies about pollution around the UK from our previous three speakers. So I was going to sort of zoom out and take a sort of global look at what are the most severe pollution problems in our different ocean basins around the world. And obviously, none of these pollutants or these factors that affect marine mammal health produced by human activities work in isolation. So they're all present in all the oceans. But when we look at different ocean basins, and I'm highlighting here as in red as I talk, we're more concerned about some specific pollutants in some places than others. So this gives me an opportunity to give a sort of whistle-stop tour, 10 minutes of all the pollutants around the world. And I'm actually not going to spend much time on PCBs in the North Atlantic because Rosie did such a good job focusing in on them. So I'm going to start up sort of at the top of the world in the Arctic. And here we're really concerned about mercury, most of all. And again, throughout this, I'll say we're always concerned about all of these factors. But mercury, as Rosie told us, is distributed around the world through atmospheric transmission of emissions from anywhere in the world, so it could be China, Russia, the US, and these fumes can then become deposited in ocean waters in the Arctic from fuel burning and industrial processes anywhere, and then as they're deposited into the water, mercury can be metabolized by bacteria, converted to methyl mercury, and then it accumulates up the food chain and can end up in the liver of both marine mammals and people. So the reason we're most concerned about the Arctic is not just does a lot of this mercury get moved up to the Arctic through climate and atmospheric movement, but also in the Arctic, people depend upon marine mammals for their culture, for food, and so they are actually eating the liver of these marine mammals. So they're being exposed directly to that liver in marine mammals, so we have a problem both for the marine mammals and for the native peoples who are dependent upon them for their way of life. In the middle there, you'll see a picture that's taken from a UK stranded animal. I have to make sure we always tie it back to Britain. And that was a dolphin called Spike who stranded in the River Tay. And those are pictures of timonite crystals in the liver that have methyl mercury in them. So mercury itself, once this methyl mercury gets into the liver of either people or marine mammals, it can affect the immune system, it can have neurological effects, it can immunocompromise animals. So because of these effects, there has been an international convention, the Minamata Convention, developed to try and minimize the impacts of mercury on humans and the environment, and also obviously for marine mammals. And that has now been ratified by 70 countries. Drs. Kershaw and Hall, who are two scientists based at the Sea Mammal Research Unit in Scotland, did a recent review of levels of mercury in cetaceans. And on the left, you'll see a graph that shows the sort of amounts of mercury. And on the right, a lovely graphic that shows us sort of the effects that mercury exposure can have on health systems in marine mammals. So it affects brain, liver, it can affect gene function. So it really has a suite of effects. And you'll notice in the middle of the graph on the number of the levels in different species, there's a dot way up high, and that's a killer whale. And it reinforces the point that Rosie made, that these killer whales that are really at the top of the food chain, so you have killer whales that are eating marine mammals, so they're the really top, and anything that accumulates in fat or up the food chain is at highest levels in killer whales. So these are species we're very much concerned about impacts of any fat-soluble pollutant in ocean waters. So moving south from the Arctic, we'll jump down to the Mediterranean. And here, probably the most important problem that we think about daily is impacts of sound, or sound pollution. And Nikki gave us this overview about how we have these different sources of sound. Shipping is everywhere. That top map shows noise from shipping, well, the shipping routes around the world with the hot spots in red and then cooler spots in blue. And so you can see there's some major shipping routes which actually overlap with the distribution, not only of cetaceans, but also of their migration corridors. And then in addition to shipping, we have sonar produced as an effect of military activity, and also for offshore oil and gas exploration, where to sound the sea bottom and detect changes in the seabed, we use sonar to look for those changes. And this has really dramatic impacts on marine mammals, because that impulsive noise can disrupt a whale when it's doing its decompression dive. So instead of coming to the surface slowly and then doing a series of small dives and getting rid of that excess gas, that gas can cause bubbles, and they can get basically the same as decompression sickness in people. They strand because of these gas bubbles in brain or in their arteries and vessels. And this occurs regularly within the Mediterranean, in the Hellenic Trench. There are a lot of examples of beaked whales and deep diving whales, sperm whales, stranding around the coasts of Greece. But most of what we know about gas bubble effects in marine mammals has actually come from work on the Canary Islands, just outside the Mediterranean, associated with NATO exercises that were using sonar as part of the exercise, and that resulted in strandings of beaked whales. Fortunately for us, not fortunately for the whales, but very close to the vet school in Las Palmas, where another of our heroes, Antonio Fernandez, on the left in this picture, was a pathologist, and so he was able to do really detailed examinations of these whales and identified the gas bubbles, and pretty much blew the science out of the water that marine mammals couldn't get the bends, and we now know that they can. And this work, he then continued over 20 years and worked very closely in conjunction with our hero, Paul Jepson, photographed here, who really led the development of the stranding program here at ZSL. And the two of them continued to work on doing postmortems on these beaked whales, but they were also really active in policy, as Nikki has reminded us of the importance. And this led to a ban on the use of sonar around the Canary Islands. And since that ban, there have been no further mass strandings of beaked whales associated with sonar. So the last beaked whale mass stranding associated with at all was in 2004, and after that we have this lovely example of success of learning something from strandings that's resulted in a policy that is now protecting animals. And I have to say that it's not only just up to people and policy. Sometimes the whales take matters into their own hands. So there is a small pod of killer whales off the Straits of Gibraltar that have taken to attacking boat rudders. Nobody knows why, but I think we can imagine why they're doing this. And they have actually sunk a number of small vessels by attacking these boat rudders. So now moving further out west into the North Pacific, again, I'm going to reiterate some of what we heard from Sarah, but we have these huge amounts of marine debris in the Pacific Ocean, and there's one that's really quite famous because of the extent of it, the Great Pacific Garbage Patch. And this is over 1.6 million square kilometers of plastics, which is mostly discarded fishing gear, but it's also plastic bottles and just about anything else that you can imagine tossed off the shores of North America, Japan, Russia, China, ends up in this great Pacific garbage patch. And this patch of all this sort of spinning debris is just north of the Northwest Hawaiian Islands, which is home to the endangered Hawaiian monk seal. So Hawaiian monk seals are endangered. There are about 1,600 in the population. And over the past 50 years, there's been a really detailed monitoring program with volunteers going out to the islands over the summer and monitoring pups being born and then helping on disentangling any animal that's entangled. So since 1972 to 2022, over 400 animals were entangled. But then in the 90s, a really big effort was begun to go out to the islands and remove any of the plastic and all the marine debris that was around these islands. And that picture there is of someone pulling up a mass of entanglement and of nets and just rubbish. And in the middle of it all was a monk seal entangled. So since the 90s, over 945 tons of marine debris have been removed from the reefs. And then since then, if we look at the entanglement rate for monk seals on the different islands, before this removal effort started, are the bars in blue, and then post-removal are the data from the entanglement rate for seals in red bars. And this is work done by Jason Baker and colleagues in Hawaii. And so they've really demonstrated that these sort of heroic efforts to remove all this stuff has actually resulted in increased survival of monk seals, decreased entanglement. And in fact, in just last month's new update of the IUCN Red List, monk seals have been downgraded from, in a good way, from being critically endangered to threatened. So we actually have a recovering seal population. Going a little bit further south into the Indian Ocean, a lot of the same problems, but here we're really concerned about some of the emerging chemicals that we don't know much about. And this is because there's a huge drive there to use new herbicides to protect rice paddies, and also insecticides to combat some of the really nasty insect-borne diseases like dengue and malaria. And just recently, we've been doing some work on river dolphins in Cambodia and have found a compound called oxadiazone in blubber. And this is the first detection in marine mammals. We have no idea what it does, but it's used as a herbicide on rice paddies. And I think this example really draws attention to the point that Rosie made that, we shouldn't be waiting to find out what a chemical does to a marine mammal. It takes years to get the data to document effects. We need to be proactive and proving that a compound is safe before we put it out into the world and it accumulates in our food chains. But we're not like that. Then jumping down to Antarctica, here we have a rather different example. And somebody asked about coastal pollution. And we have a term that we use in the infectious disease world of pathogen pollution. And this essentially means that things like viruses, bacteria, protozoa, worms, parasites can become pollutants of coastal waters when they run off from land and when terrestrial animals that normally harbor these, including people, that runoff can go down into the coastal waters, and then we get marine mammals being exposed to parasites and infections that they've had no previous exposure to. So they haven't got the right immune systems, they don't recognize these parasites, and these naive populations can really be very vulnerable. So we have a very extreme example in Argentina just a couple of years ago, when H5N1, the new avian influenza variant, killed 17,000 southern elephant seals in a colony in just a couple of weeks. So we're really concerned now about Antarctica because we have huge populations of seabirds as well as marine mammals, and influenza has just reached the Antarctic Peninsula in March 2025, and there have been reports of seals. And so this is, I think, a real example of thinking about pathogen pollution. And here again, always trying to get a little seed of hope. I think a lot of the tourist vessels and cruise ships now are really aware of this risk and are starting to institute quarantine procedures for people visiting the Antarctic. Moving back up towards England, in the Gulf of Mexico in the US, we have probably the most extreme example of oil spill pollution, where in 2010, the Deepwater Horizon oil spill was the largest oil spill in history. So it was absolutely devastating. There was a human loss of life as well as hundreds of marine mammals. And I think that oil spill really taught us about the acute effects of oil on marine mammals, how it can basically swamp them, stops them from breathing, comes up on their skin, stops them from being able to swim. But we also realized by monitoring after this event that there are chronic effects that can happen. So oil fumes can be breathed in by dolphins. They can eat prey that have been contaminated by oil. And this leads to long-lasting effects on liver function, on blood function. And so we now see 10 years later, we're still seeing stranded dolphins with impaired immune function, damaged kidneys, damaged lungs because of the effects of that oil spill. And then our final example is an example really of a problem that is becoming really exacerbated worldwide, these harmful algal blooms, or red tides as they're referred to. And these are increasing in frequency, in distribution and duration and toxicity worldwide since the 1980s. And most of this is associated with climate change. But pollution, i.e. coastal runoff of nitrates from mostly agricultural use, make these coastal waters more eutrophied. There's more nitrates. This encourages growth of these algae. And so they produce these toxins that again are transferred in the food chain, very much like the chemical pollutants, and can cause acute death. Animals, some of these toxins cause seizures, they can cause reproductive failure, and they can also cause chronic organ damage. So on the right, I have an example of the biggest bloom that we saw in California on the west coast of the US recently. In 2015, there was a harmful algal bloom along the entire west coast of the US. And the top graph shows changes in wind speed, in ocean temperatures, and then the algal response to these changing oceanographic conditions. The bottom is a map enhanced for chlorophyll, so you can see the red is where there's this red tide. And the pictures on the left are California sea lions piling up in the marine mammal rescue centers where they've been having seizures and they die and they have abortions. In the same year, down in southern Chile, the greatest number of whales that recorded stranded ever were associated with a harmful algal bloom in that same year. And down in southern Chile, there were over 300 sei whales documented, washed up, dead. And it was a hard stranding to investigate, a very remote area. But a lot of sort of sleuth work by Verne Hauserman and her team identified an Alexandrian bloom that produced saxitoxin associated with these strandings. And saxitoxin was found in a couple of these very long dead whales. So this is an area we think some of that eutrophication is not so much coming from agricultural use, but use of compounds around fish farms all around southern Chile, where there's more pellets of fish food to feed the fish that then leads to proliferation of blooms in those areas. So that really was sort of the big whistle-stop tour of the oceans of the world, and with these examples of really extreme problems in different places. And I think when you look at this, it's really easy to get depressed about the state of our oceans and what are we doing to them. But I think at the same time, we really have to have this little glimmer of hope. Each of our speakers gave us examples of where we are able to do something. So I think my take-home message to you is that by looking at stranded animals, we can identify what the impacts our human activities are. And then if we take that information and we share it with managers, policymakers, they can do something. They can pass laws, they can develop policies to minimize some of those impacts. And then we, as citizens, we can make choices about what we do. Rosie told us how we can make choices about what we eat, where we shop, what we do with our stuff. I have to say that we can vote for the right policymakers that will do things that will support what we care about. So really, I think there is hope. We've heard about monk seals are now increasing because of efforts to clean up marine debris. PCBs are decreasing in the North Sea and in the Baltic because of efforts to reduce their use and their discharge. And then we have examples of no more big whale stranding in the Canary Islands. You know, Antonia and Paul are out of a job. So there are some really great examples of how we can do something. So with that, I actually really want to take this sort of platform up here where I can talk to you and many others online to really thank everybody who works with the stranding networks. Most people who respond to strandings are volunteers, many are students, and as supervisors of students, we often abuse them and make them work hard in return for their ill-gotten gains. So I just really want to thank everyone who works for the stranding networks and with stranding networks to make their data valuable and with use. And then, of course, I have to thank Rob for the invitation to be here. So thanks, folks.
Thank you, Frances. Thank you very much. Don't go away. Hang on. So we didn't drink it all last night. There is still a bottle of California Pinot Noir left. So thank you very much. We are slightly over time. So perhaps while you're getting all the rest of the speakers back up on the stage, please have the lights up again. And then thinking about questions, particular questions for Rosie and Frances. I really didn't allow a question in their talks. That would be great. So if you have the lights up, thinking about questions both in person and online, that would be great. So let's go for it. The gentleman at the front, I know you had a question earlier on. I don't know if I'm asking anything now.
It's really about the military sonar and the stranding of all those big whales on the Atlantic coast of Scotland and Ireland. It's widely speculated that it's Russian activity looking for pipelines across the Atlantic that may have caused that. I just wondered what thoughts you had on whether it could have been military sonar.
I mean, obviously I talked a little bit about the potential for links because, it's no secret that military are creating impulsive sounds during activities. The military, the UK military particularly, they do work quite closely with things like the Strandings programmes and other experts to try and understand what the impacts could be, and the modelling, like the drift modelling that I talked about, helps us to get an idea of whether there are potentially connections with activities that are creating impulsive sounds. But there's no real conclusive way of knowing what the links are, and when you're talking about something that's linked to national security, then there's obviously a whole host of other things that you need to consider and will potentially take precedent over, sort of the welfare aspects. It's quite a difficult balance to strike when you're dealing with national security. But we don't have all the details of what's happening in the military. They obviously have things that they can't divulge, but they do work really closely with quite a lot of the experts to try and make sure that they are communicating and engaging as much as possible.
If I could add something to that as well, Nikki, if that's all right. Yeah, we do have really good collaborative relationships with the Royal Navy now, which are great, but also to point out in that area it could be multiple navies having multiple activities, and therefore it's hard for us to gather that information as well. So it's better. It could be better still, but that's where we are. Thank you. Any questions? Linda's got a question at the back.
Thank you all for really great presentations. My question is for Rosie. I was really impressed by the 190 milligram per kilogram in that killer whale of the PCBs, and you said that the suggested threshold level is 9 milligram. And my question, probably obviously, is how is that threshold informed? Where does this come from? And is that for killer whales or cetaceans in general? And how, where does the suggestion come from?
I'm really glad you asked that question, because if I had time, I would have wanted to cover it. So these thresholds are really the best we have in lieu of something more robust. So I think it's important to take them in the context of how they're derived. So the 9 milligrams per kilograms is across all cetaceans, and I think maybe marine mammals as well. So obviously there's a huge breadth, as we've heard, about the different cetaceans, marine mammals, and biology. So they're likely to be affected in different ways. It's derived from a mixture of in vitro laboratory studies, also surrogate species, so other species that are able to be in a lab environment and exposed to pollutants. So often things like mink are used as surrogate species. And then there's some extrapolation between what you see, these thresholds in laboratory, both in vivo and in vitro. And then that's extrapolated out to marine mammals, also from some feeding experiments that were done in the 1980s, and this magic number is produced. So I think it's really important to take it in the context. It's deemed as the threshold for the onset of physiological effects. That again is really broad in terms of that definition. So I think it's likely in killer whales they're able to tolerate higher levels, but whether they're able to tolerate levels that are that high is a big question mark. So I think it'd be really great if we can derive more robust thresholds, again, because a lot of this work, so that threshold's from 2012, I think, off the top of my head, I need to check. But yeah, I think there's lots of innovation within that space to try and develop more robust thresholds.
Thanks, Kata. Thanks, Rosie. Can we go for a question online, perhaps, and then one of the audience, sorry, if that's okay?
I had a lot of praise from everybody watching online, so thank you all very much for your presentations. They were fantastic. We do have quite a few questions online, so if I may do a couple, but I'll start with one. Do we know if micro and nanoplastics can be transferred across placenta or in milk from mothers to babies?
Sarah?
Yes, we do. They can. Yes. So we don't know in marine mammals, but we suspect they do because they've been found too in humans. So microplastics, nanoplastics are being found in human breast milk, placentas, testes, basically everywhere you look, they're there, which is a bit terrifying. So.
Thanks, Sarah. Another one, Harriet?
Thank you. Do you think, I guess this could be to any of you, do you think that marine animal watching activities are inherently damaging to the animals? Is there any way they can be done with no harm to the animals, either in terms of noise or habituation, etc.?
Who wants to go for that?
I can start that one.
Okay.
I mean, there's very different ways of going about it. There's a lot of really good guidelines that exist that have been created at different levels, either from government or created by the industries themselves, that give really good criteria to make sure that your impact on those types of activities is minimised. It obviously depends on people following those when they're interacting with animals. There are instances where there's harassment problems, where animals are continually being pressured by tourism boats because it's sort of a one after the other approach. But then there's also examples where it's really well managed and really well handled. So it very much depends on the situation and how well these guidelines are stuck to as to whether there's a significant impact or not. I don't know if anyone else.
I might just add a point. I think the question was about inherently bad. I would say not inherently. If you can watch an animal in a way that does not disturb it or alter its behaviour, I think that's the key. And then I also think that, obviously, we're all mammal fans because we're here, but there are people who don't know much about marine mammals and they have to make a living. And so if we can share our passion for marine mammals by allowing other people to see them, that has to help in the long term. So the onus is also on us to find a way to allow people to see marine mammals and give them guidance on how to do it without altering the animal's behaviour or upsetting them in any way. And there are some great guidelines. The International Whaling Commission has some guidelines on their website, and a lot of the industry themselves have their own guidelines in different countries.
Thanks, Francis. Do you have a question here as well?
Sure. So, thank you all for the fantastic talks. My question's for Rosie. You talked about how there's transfer of PCBs from mothers to calves during lactation. And do we know how quickly those PCBs build back up, or is there a greater impact on the first calf compared to subsequent calves?
Yeah, we do. So there's a much greater impact on the first calf. If the mother is able to successfully reproduce and have that first calf, if the calf doesn't survive but they've lactated for long enough to offload their pollutant burden, then there's much better chance of survival for the second or third or subsequent calves. The impact is really if PCB levels are so high or other pollutant levels are so high that they're not actually able to reproduce, then they're not able to offload their pollutant burdens. And in terms of it building back up, we don't see, when we look at the Strandings data, we see the adult males have the highest levels. So we don't see that actually adult females accumulate as much. If they've been able to offload them, it seems to stay that they have relatively lower levels of pollution than other age classes.
Thank you very much.
Thanks, Bert.
Yeah, thank you all for the education. They were really great talks. It was mentioned in a couple of them around the sort of ghost gear. And I know there's kind of some policies that have been put in place across the world to try and kind of cut down on the amount of that that's thrown into the ocean, but I imagine it's very difficult to enforce. So yeah, just out of interest, do you know how we're trying to enforce that in the UK at all?
Kind of action, I guess, is cleanups, so beach cleans and things like that. Although it sounds quite piecemeal, actually we have a very engaged population of people who like to clean beaches. And the UK is one of the kind of best countries for beach cleaning. And there are other policies and interventions, such as, so some harbours have sea bins, or bins that fishermen can bring back ghost gear that they find while they're fishing, and they might collect it in their own gear, they'll bring it back, and they don't have to pay to dispose of it, which in other cases they do have to, so there's the incentive to not recover a ghost gear then. There's also the potential to use kind of ropeless gear, which is currently in development. There's a lot of trials going on at the moment. Obviously, there might be implications for fishers in terms of the efficacy to catch fish. Plastic is a very useful material. There's a reason why it's so prevalent. And it's obviously in the early stages now of trying to switch to a more kind of environmentally friendly alternative, but there's a lot of work to do on that.
Thank you both. Gentleman in the middle. And then to Harriet for an online, if that's okay.
Really great talk, guys. Thanks for all your words and your time. I guess it was kind of a point similar to this gentleman's, which is actually there's a lot of great data that you guys have got. But what's the I guess the way to pressure government and maybe through public education, which I guess is maybe lacking in some of the depths of these subjects, to sort of make some of these things happen a bit more quickly, because as you guys said, it's sometimes quite reactive and takes a long time to create action, but it feels like action is kind of needed quite soon.
Who would like to handle that one?
It's got to be you, Francis.
I'm going to toss that back to Francis, maybe, if that's all right.
Coming from a dark place.
That's what I want.
Let's not go there, just in general then.
There is a new high seas treaty, just a new convention, ready for ratification.
Okay.
Maybe, I don't know.
I was going to touch on the plastics treaty as well.
Yeah,
I mean, this is an example of a thing that should work, and it's been going across rounds and rounds and rounds of negotiations, and we're still not there, because there are lots of people who don't want it to work. There are lots of plastic kind of lobbyists at these events that outnumber scientists, actually, and their voices are very loud. And they, making all these countries from around the world agree on even just the text of these pieces of legislation is proving very difficult. So yeah, it's a tricky one.
I'd probably say from a kind of UK perspective as well, it's a lot to do with how information's collated and presented as well. I think, really clear messages, really clear links. It's a lot easier, because the kind of, governments are bombarded with decisions and things that they have to try and weigh up and balance. So if you can provide really clear messages, particularly, through Strandings programs that have been running for decades, when you've got, that time series of information and some really good links, positive actions, solutions, then you've got a much better chance of having those conversations that then have that impact.
From a chemicals point of view, there's similar challenges, but something that I really care about at the moment is the lack of consumer choice we have around the globe, but certainly in the UK, and that you know about these chemicals. If we take flame retardants as an example, or sort of waterproof clothing, we know that a lot of these chemicals are toxic. It's impossible to go into a shop and say what chemicals are in that waterproof jacket, what flame retardants has that mattress been sprayed with, or that sofa. As consumers, it's really difficult for us to make the right choice. So that's something that I'm trying to think would be really useful to push, because then you have the education piece, and then if consumer choice changes, industry is forced to change. But at the moment, we're going in blind. So I think that's something that would be really important to try and push.
I think we'll perhaps have one more from Harriet, and then we'll wrap it up. I was about to say no, but she's looked at me sadly, so I said yes. Rubbish tearing, eh?
I'll make it a really quick one. I've combined a few into one question. There's been quite a few questions around actually collecting data and sampling. So does decomposition condition of stranded animals affect how you collect toxicology samples? Are there any other ways to collect these samples, perhaps before they're stranded? A quick one.
Sure. And yes, decomposition does. As you think about Rosie's measurement, it's milligrams per gram of fat. And as animals decompose, the fat drips out of the blubber. Not a very pretty scene to imagine before you go out for dinner, but it does. And so decomposition does affect the measurements. There are quite a few projects around the world where live animals swimming are biopsied. So you can fire a crossbow dart, and then that'll take a plug of skin and blubber from the animal. The dart will fall out into the water, and then you can go in a boat and pick up the dart, and the animal swims off. And I should say there's plenty of follow-up observations to make sure that those little dart holes heal and that the animals aren't impacted, so it's not impacting their health. So those biopsy studies have told us a lot about PCBs and contaminants in animals that don't strand. And there also has been quite a lot of work done on bottlenose dolphins in Sarasota, Florida, where the animals are actually caught at periodic intervals every sort of couple of years. And they're given a health assessment and a blood sample taken. And PCBs have been measured in those, and those animals are caught and then they're released again, and then they know all the animals, so they've got 50 years worth of data on mothers and calves and all their health problems or not, and how well they do, combined with blubber levels of their contaminants. So that's not affected by stranding or decomposition or anything. And we know from all that work that animals with higher levels of PCBs in their blood have poorer calf survival and poorer immune function.
Once again, thanks to all our wonderful speakers. I'm just going to give a few last slides. I don't want to shine it in your face. Maybe you guys take a seat. That's all right. And then we'll wrap up. Can the slides back up again, please? Okay, so this is a really obvious point, and it's already been well made by everyone else here as well. Stranding networks rely on a huge number of people to help out. Some are in the audience today, many are online. So from my end, I want to say thanks and huge gratitude to everyone who helps us out with getting bodies off beaches, post-mortems, and bringing us either cups of tea or a pint down the pub later on. So thank you everyone who helps us out. I want to say thank you to some key individuals who've helped the programme out over the years. Some are very much still with us. Unfortunately, some, however, are sadly not. So again, huge thanks to them. I want to end tonight, though, by paying particular thanks to our good friend and colleague, Dr. Paul Jepson. So you've all heard tonight how Paul's work has fed into most of the research here tonight. This is Paul at work. This is Paul more naturally at work. He ends up covered in claret on the beaches. So he's been involved with and running CSIP for nearly 30 years. I think this is really Paul in his more natural habitat. He loved teaching. He was a fantastic teacher in communicational science, and I learned a huge amount from him personally. So, unfortunately, as many of you know, Paul's had to step away from work a few years ago now. He's on a personal journey dealing with the impacts of Huntington's disease. So I want to close tonight by paying tribute to Paul's key role within CSIP over nearly 30 years. The work continues, but we're only here because of his efforts. So I know Paul and his wife Vanessa are watching tonight, or at least I hope they are. So for everyone in the audience and anyone online that knows Paul, please send them both your love and hugs, and please raise a glass to them of your favourite tipple tonight. I certainly will be. So many thanks to Paul. And thank you all for attending tonight. I do have a few, before we start clapping, I hope you clap. I've got a few. I have some final housekeeping and then one last message. So once again, thanks to my wonderful friends and speakers tonight. Fantastic talks, thought-provoking presentations as always, and thank you all of you for getting involved. I appreciate we ran out of time, so bear with us. We'll try and get back to you if you have any other questions. The next Science and Conservation event is going to be on the 4th of November. We'll be looking at genomic health and reproduction in conservation. Please check the events page at ZSL website for more information. Let us know what you think about tonight's event. Again, nice thoughts only, please. That's what we like these days. And final message for tonight. I want to thank you all for listening. Huge thanks to Harriet Macara, who runs things fantastically capably here. Wouldn't happen without her. And for her much appreciated support. Thank you again. Safe travels home tonight. Look after each other. Spread some random acts of kindness wherever you go. Make the world a better place. And we'll see you all on the beaches again soon. Thank you, everyone.